The chemical digestion of food is performed within the alimentary canal, specifically the mouth, stomach, and small intestines. Enzymes, which are protein-based selective catabolic agents, play a major role in the chemical digestion of food. The selective nature of an enzyme is based on the active site shape, which only allows specific molecules to dock. The molecules that dock with an enzyme are known as substrates. Once a substrate docks, the enzyme causes a hydrolysis reaction to occur, cleaving larger bonds of the food into smaller portions. Because of the specificity of enzymes, many types are required for the digestion of the three classes of macronutrients. Carbohydrates are broken down from larger molecules known as polysaccharides, which consist of starch and glycogen, by the enzyme amylase. The breakdown of starch into the disaccharide maltose is the product of this hydrolysis reaction. Maltose, which is a combination of two glucose molecules, is broken down into monomers of glucose by the enzyme maltase. The other types of disaccharides are sucrose, which is broken down by sucrase into fructose and glucose, and lactose, which is broken down into galactose and glucose by the enzyme lactase. Lipids are broken down into glycerol and fatty acids, and proteins are broken down into amino acids.
Where does it all begin? For carbohydrates, chemical digestion starts within the mouth as the enzyme salivary amylase is secreted from the parotid gland and mixes with the food as it's being chewed. As the bolus is formed and then swallowed, it passes through the esophagus to the stomach. Once the food reaches the stomach, the salivary amylase becomes denatured due to the low pH levels of the stomach. Chemical digestion of carbohydrates briefly stops here before moving to the small intestines. The hydrochloric acid of the stomach activates the pancreatic amylase to be released into the duodenum of the small intestines to continue the digestion of carbohydrates after passing through the stomach. Pancreatic amylase finishes the work of salivary amylase and converts the polysaccharides into oligosaccharides and disaccharides such as lactose, maltose, and sucrose. The oligosaccharides and disaccharides then encounter the brush border enzymes where they are converted into monosaccharides, glucose, fructose, and galactose. The brush border enzymes are located within the cilia of the microvilli and initiates the final step of the chemical breakdown of carbohydrates. Enzymes such as dextrinase, glucoamylase, lactase, maltase, and sucrase are used to complete the breakdown of disaccharides to monosaccharides. Glucose is the major molecule of this breakdown and that is used in many biological processes, especially in ATP production.
When proteins reach the stomach, the chemicals have already been prepared for digestion. Prior to ingesting food, gastrin is produced by the G cells in the stomach. Gastrin will activate the parietal cells of the stomach to produce hydrochloric acid, which is required to form the enzyme pepsin. Pepsinogen, the inactive form of pepsin, is secreted by the chief cells of the stomach. When acted on by hydrochloric acid, pepsinogen takes on its active form, pepsin, for the initiation of protein digestion. Pepsin requires a highly acidic environment to work within. As the proteins are digested both chemically and mechanically, they are moved towards the pyloric sphincter. The pyloric sphincter will open to allow the chyme, a mixture of partially digested food, to move into the duodenum of the small intestines. Once in the small intestines, pepsin is deactivated due to the sodium bicarbonate that neutralizes the acidic pH of the chyme. The next set of protein digestive enzymes are secreted from the pancreas. Trypsinogen, the deactivated form of trypsin, is secreted into the small intestines by the pancreas and is activated by the enzyme enteropepsidase, found within the mucosa of the small intestines. Trypsin combined with chymotrypsin and carboxypeptidase denatures the proteins of the chyme further into their smallest components, which are amino acids. Amino acids are absorbed through the wall of the small intestines through cotransport and travel to almost every part of the body for protein synthesis. Fat digestion, similar to carbohydrate digestion, starts in the mouth with the enzyme lingual lipase. Although this enzyme plays a minor role in digestion, it is still important to note. As fats enter the stomach, gastric lipase acts on the fats and again plays a minor role. Once fats enter the small intestines, the true chemical digestion begins. Bile salts created by the liver are secreted into the small intestines through the common bile duct to emulsify large fat droplets. Bile is required for the digestion of fats due to the watery environment of the small intestines. Since fats and water do not mix, the bile salts enable emulsification, which causes the large fat droplets to be broken down into smaller droplets, which are easier for enzymatic digestion. Pancreatic lipase, the major fat digesting enzyme, converts triglycerides into monoglycerides and fatty acids. Once the fats are broken down into their smallest components and are combined with bile salts, they can be utilized in the body to build phospholipid membranes, make energy, and assist cellular growth.